You guys are troopers. You've made it through. So now that we've gone through a whole bunch of stuff about biochemistry and receptors and enzymes, let's talk about drug discovery and development. And this is important now because of the whole COVID outbreak and drugs that they're trying to develop to take care of people and vaccines and things like that. I'm going to focus more on drugs, things like aspirin and those types of things, but we can also talk a little bit about vaccines because they do follow a little bit different process. But this is the process. This is probably going to be a couple of videos. And again, you can go through the slides on your own. And obviously this is doing it very quickly because I've taught an entire course on this before, so there's a lot more to it, but this is a very good overview. All right, so first of all, the agency that regulates this is the Food and Drug Administration, and the bottom line is for a drug to be approved, it must be safe and effective. Now, safe is a relative term because there's no such thing as a drug with no side effects. So safe can be any number of things. So if you are a cancer patient and you're going to die if you don't get a drug, and the drug makes you really, really sick, well, that's safe enough for something like that. But if you have a headache and you take an aspirin, right, the side effects are very different. So safe is a very important term, but it is a relative term. Effective means it actually does what it says it does. So if it's supposed to make your headache go away, it should make your headache go away. If it's supposed to kill cancer cells, it should cure cancer cells. And all these decisions are made on a large amount of data. And one thing that's not really in the slides, but that threads through is the FDA is not involved at the end. The FDA is involved through the entire process. There are all sorts of checkpoints along the way that they have to send. A company that's trying to get a drug up for approval has to send stuff to the FDA at various stages to get approval. The way all the testing goes follows certain sets of regulations, and there's a lot of detail in that we're not going to go into, that is designed to ensure that the data means something, that drugs are developed with a set of regulations, and that no one's cutting corners. Obviously things happen, but in general that's how it's supposed to be done. All right, this website is fdareview.org, and this has a kind of a nice overview that you can look through on your own, and you can go to this website if you'd like. This is from Pharmaceutical Research and Manufacturers of America. This is in general the timeline. So you can see this is, once it gets into preclinical testing, it still takes about 15 years to get a drug approved. Right? And this is a nice summary that I found. And it costs a lot of money. It costs about $2 billion to get a drug through the process. And that doesn't count drugs that didn't make it. That is just drug A that made it all the way through. 10 to 15 years and about $2 billion. It's a lot of money that a company is investing to get it back. All right, so the first step in discovering a drug is you have to know the biology of the disease. So if you're looking at something like, say, diabetes, you have to know how diabetes works and what causes it and what's going wrong. Or if somebody has say dementia, what's going wrong in dementia, or any other disorder, a cancer, things like that. A lot of it, so you might want to review, you just went through it, but the receptor and enzyme slides, typically in a disease, there's something wrong with that process. There's some signaling mechanism that isn't working, or it's working too well. It's an enzyme that isn't working right. And of course, if you're talking about an antibiotic or an antiviral, you have to know the life cycle of the organism or the virus that's causing the disease. And then what you do is you determine what the receptor or the enzyme is not working. That's referred to as the target. And then you develop an agonist or an antagonist. And to do that, you have to have the structure of the target. So this was the last, or one of the last slides on the receptor enzyme slides. And this I pulled from Journal of the American Medical Association. And this is the process that the COVID virus gets into a cell. And all of these little boxes here with these little arrows and the words in green, the words in green are drugs that are being worked on, and this is the target that it's after. So camostat mesylate inhibits TMPRSS2, preventing viral entry. So that's the target for that drug that's being developed. Arbidol targets this protein, and so on and so forth. But you can see various stages. You have to know the life cycle, and every single thing, every step is a potential target. And so drugs can be developed to go after any number of targets. Right? If you want to look in detail, this again, this isn't really a slide to look at, but you can read through this. But these are various drugs that are out there that are being worked on right now, specifically what the target does, toxicities, all sorts of stuff like that. And that's something you can look at on your own. And this was as of April of 2020. All right, so let's start with drug discovery. I'm calling it a fishing trip. So basically, once the target is identified, this is the molecule that we're going to try to fix. We determine the structure. And then the first step, and this is relatively new, is in silico design and testing of potential molecules. So in silico literally means on a computer. So there's some software that will help you draw, literally like Lewis dot structure, but they can deal with it in 3 dimensions. So Lewis dot structure in 3 dimensions, draw molecules that will fit into the pocket. If you remember the lock and key hypothesis or hand and glove hypothesis, draw molecules literally with a computer pen that will fit into the target, into the active site. And then they do docking studies, and they go, okay, this will fit. Maybe if we put a chlorine here, this will fit better. If we add a 2-carbon group here, it'll fit better. And try to design stuff in silico that may or may not work. It actually saves a lot of time because you don't actually have to make it. All right, once there are some drugs that in silico look like they'll work, then it goes to organic chemists and they develop a synthetic program and they make a library of compounds. And just like the word library, you literally might make 3, 5, 10,000 molecules that are variations on the same theme. So they're going to be similar structurally, but one may have, as an R group, a 1-carbon molecule, maybe a 2-carbon molecule, maybe a branch chain, maybe a fluorine or a chlorine or an oxygen sticking off. And they'll literally make families of molecules and literally in test tubes make thousands of molecules. And they're actually going to do this literal testing. So this is actual molecules, not in silico, right? But they have to. It's a target. So you have from in silico, you go stuff that looks kind of like this might work. And then of course they do in vitro testing. So in vitro means in a test tube or in a flask. And typically what they do, what's done is now we've made this 1,000 molecules. Let's say it's 1,000. And then we have the receptor that we've purified, and we have a whole bunch of it. And then we say, does this molecule actually bind to the receptor? And we have 1,000 of them. And so we may, well, of the 1,000, how many of them actually bind to the receptor? Anything that doesn't bind, throw it away. All right? Once stuff binds to the receptor, then we're going to try maybe in cells, and then we'll try binding it in tissue and seeing what the next steps are. So you work your way up. All right? So an in vitro fail, and actually economically, failing early is better, right? Because you don't have to spend as much money. Anything that doesn't bind to the receptor. If it binds to the receptor but doesn't do what it's supposed to do, okay, that would be a fail. Toxicity, right? So a toxicity meaning it binds to the receptor but it makes the receptor break, or it binds when you get to the step of it's working, you're doing the receptor, but actually in cells it kills the cells if that's not your goal, right? Or causing damage. Right? So the hierarchy of in vitro testing is first we do the receptor, then we do cells. So the receptor is a purified molecule, but that molecule lives in a cell. It might be on a cell surface, it might be inside of the cell. So then we'll do actual testing with cells because it might bind to the receptor, but it doesn't get into the cell in the first place, okay? Or it might kill the cell. Once it's cells, then it's tissue, because tissue has more than one type of cell. And then maybe even slices of an organ, like liver slices or something. At each stage, drug can fail. Toxicity is looked for at each stage. And you might start, if you start with, say, 5,000 or 6,000 molecules, you may have, I don't know, several hundred that go through that work in this stage. All right? So once we go through that whole process and we get some molecules that look like some drug, some potential drugs that look like they're going to work at this stage, all the in vitro testing says it looks good. Then we do in vivo testing. And in vivo, vivo means living. that means you're testing in a living thing. So I'm going to talk about this briefly, and then I'll go to another, I'll make a second video. All right. So in vivo is in a living organism, and specifically it's animal testing. Now I know a lot of people have problems with animal testing, and no one wants to kill an animal needlessly. But the law right now is that drugs have to be tested in at least one rodent and one non-rodent model. So, mice and rats and then some other animal. The reason we use animals is because even though computer modeling is very good, there is not in existence a computer model that is an accurate simulation of what happens in a real organism. And that's because I think most people don't appreciate the complexity of a living organism. When something goes into a human or a mouse or anything like that, there's a lot of other variables that come into play. And until such time as there is a really good computer model for that kind of testing, it's going to be in animals. Okay, like it or not, the animal rights movement has done a lot, though. Animals are treated much better. There's a whole bunch of laws around that right now. So, but bottom line is it goes into animals before it goes into humans. All right, I'm going to stop this video here, video number 1, and then we'll go into video number 2.